Hello, welcome everybody. It's a pleasure to see such a big audience, especially after first evening of Hospitality Suite. So really appreciated that you make effort to get up so early. We will be starting, so if you still wait outside, please come inside. We will start in a second. So it's great to have such a big audience today. This is a seminar for launching Spectronaut 19, and I have a pleasure to chair this seminar, and I will give you a short overview of the plan for today. So firstly, I will make a short introduction to our software. Then we will have first speaker, Tejas Gandhi, head of bioinformatics at Biognosys, and he will give you overview of what's new in Spectronaut 19, focusing on the improvements of direct DIA. Then we will have first external speaker, guest speaker Josh Coon from University of Wisconsin-Madison, professor of biomolecular chemistry. And his talk is about a single-shot DIA proteome analysis as the new gold standard for the quantitative proteomics. Afterwards, we will have a second guest speaker, Denys Oliinyk, from Functional Proteomics Lab at Jena University Hospital. And his talk will be about high-dimensional phosphoproteomics with data-independent analysis and microphos. So let me start with the short overview of our software suite. Spectronaut is our gold standard for DIA proteomics, featuring such workflows as direct DIA and library-based DIA analysis. Next to that, we have SpectroDive for targeted proteomics. It supports such workflows as MRM, PRM, including PRM passive, FAMES PRM, and SureQuant. And finally, we have Spectromine for data-dependent acquisition. When working on the new software, we are focusing mainly on three pillars. First, we would like to be the best when it comes to the efficiency, scalability, and performance compared to other alternative software there on the market. Second, we would like to have a trust of scientific community, and it's our pleasure to see that every year there's a growing number of the publications mentioning our software. And finally, we would like to give you the best user experience with analyzing the data. And with this in mind, we were working also on this new release of Spectronaut. More on the Spectronaut 19 we will hear from Tejas. I would like to just mention a few key features of the software. So it does have the best performing library-free approach for DIA analysis out there with our proprietary direct DIA workflow. Secondly, it has AI in the core of development. It has best performing PTM analysis workflow for DIA analysis, and here we had also further improvements in Spectronaut 19, and this we will hear a little bit more about from Tejas. We also work towards facing the challenges of the modern proteomics with the scalability and unlimited throughput analysis. And finally, it's one all-in-one solution that you can start from your raw data and reach biological insights with the powerful visualizations and reporting. If you are interested, we offer 30-day trial licenses for all of our software. We offer also tiered pricing that has attractive pricing scheme for the academic and non-academic users. And finally, if you would like to bundle it with other our software, we also have attractive discounts for the bundles. And with this, I would like to thank you and give the scene to Tejas. If you have any questions, please hold them until the end of the seminar. We will have one common Q&A session in the end. Thank you.
Thanks. It's great to see so many people. Oh, you guys can hear me, right? Okay. Great to see such a good outcome. And yeah, I'm not sure if you can tell if I'm extremely jet-lagged. It's because I'm also extremely excited to share what's new with Spectronaut 19. So let's get into it. So every time we're planning a new release of Spectronaut, it's an exercise in balancing many different user requirements. And what we usually do is that we decide to focus on certain important themes, and at that point, everything else becomes a nice to have. So what were the main things, or let's say the 3 main goals we had for Spectronaut 19? So like Monica already mentioned, our focus was direct DIA, which is our library-free analysis workflow. And specifically speaking, we wanted to improve the identification rate of direct DIA. We strongly believe that there's a lot more we can identify in DIA than the current state-of-art algorithms. So this is something we're constantly pushing. Secondly, we recognize that direct DIA is a more computationally demanding workflow. So we don't want there to be an analysis paralysis, right? So we also wanted to really improve on that part. And thirdly, point of many DIA studies is, or discovery studies is to identify or discover differentially expressed peptides and proteins, right? And we want Spectronaut to be the best software when it comes to candidate discovery. So that was something that we really wanted to work on as well. So while the first 2 goals are relatively easy to measure and explain, I would like to say a bit more about candidate discovery. Identifying differentially expressed peptides and proteins is, well, you need 2 things. First, you need to identify them, and second, you need to quantify them well. So it's not really about identification versus quantification, as more as identification plus quantification. And what we find is that there is a rather complex relationship between all the parameters associated to identification and quantification. So while you can use surrogate matrix like identifications, right, or precision or accuracy to say something about your software's ability or experiment's ability to discover candidates, we wanted to have a more direct measure of this candidate discovery. The way, so this is a very interesting topic, and Monica has a talk about it tomorrow, so I can highly recommend you to attend, where she goes a lot more in detail than I will right now. So the way our approach to candidate, measuring this is maybe not too different from many of you, is we rely on the so-called controlled quantitative experiments. And so it's usually in these experiments, as you might know, you spec in different species in known amounts, right? And so here's a very simple example of a CQ experiment where you have 2 conditions and 2 species, and human is the control, so it's a fold change of 1, and C. elegans has a fold change of 0.67. And then you would take this and run through your favorite software, and you would get your protein fold changes, and then you have this famous or rather infamous scatter plot that people often show, right? It's a very nice plot, but the problem with this plot is that it's a very qualitative assessment of a quantitative experiment. So what we like to do and what we recommend everyone else to do is to go a step further and then do a statistical testing on this. Could be a simple t-test, right? And then you can get a ranked list of your protein candidates or peptides, and then you can basically, based on the number of true candidates, in this case C. elegans, and based on the control, you can say, like with 5% error rate, how many candidates you find, right? So you have a very quantitative outcome of your quantitative experiment. So we can use this schema to demonstrate, for example, that Spectronaut 19 performs better than Spectronaut 18 for finding candidates. But what we realized that's not enough to do this on a couple of different experiments. So we wanted to really have a large suite of this kind of experiments. And so we set out to create, that's what we did. We created the largest known collection of CQ data sets, and it covers all the different popular DIA platforms. It covers different gradient lengths, and also covers many different aspects like loading amounts, experimental design, so fold changes, number of species, number of replicates, all sorts of things. You can find, Monica will talk more about it. So now with this in hand, we can then more confidently say that actually, yes, based on a large number of different data sets, we do get better results than before. So yeah, then we know our goals, right? And now we know how to measure them. So how do we realize these goals? So there were many initiatives we took, and I would just like to talk a little bit about one of them, which is artificial intelligence. So you might not realize it, but direct AI extensively uses deep learning models, and these models can have a direct impact on identification, quantification, and speed, things that very nicely align with our goals. So we wanted to improve our deep learning models. And how did we do this? Yeah, so just have an example of 3 deep learning models that we have in our pipeline. And as you can see, we have a lot more training data. So training data is usually like, a key part of having a powerful deep learning model, especially if you have high quality training data. So we spend a lot of time acquiring this data internally, but we also use public data whenever possible. So the x-axis or the y-axis is in millions, and as you can see, we have a significant improvement in like 4 to 5-fold larger amount of training data that we have for our models. And we also, it's not working well. We also improved the design of deep learning models, so you can learn a bit more about this at the posters we have on this topic. So based on this, what were the improvements we had for deep learning models? So again, referring to these 3 deep learning models that I mentioned before, we using some key performance metrics for each of this model. You can see that there was a significant improvement of their performance compared to Spectronaut 18. Especially for deep CCS, you can see that we have like a 45% improvement in mean absolute error. And this will really help with many things like estimating the tolerances for extraction, scoring, and have a direct impact on speed, identifications, and other aspects. Yeah, so now I'm just going to go into the results we have for the goals that I mentioned. And yeah, so it's always like, you have this plot, and it's a bit of underwhelming if you have very nice numbers, because it's just 2 numbers, right? But if you didn't figure it out, we really like to benchmark on large data sets. So we don't like to say something based on a couple of data sets. So we have in this box plot, each point is a different experiment, a lot of external data sets, internal data sets, all kinds of data sets. And on average, what we see is that Spectronaut 19 gives 13% more precursors and 10% more protein groups than Spectronaut 18 with direct DIA. And we also extensively benchmark or validate FDR. And here's just one of the experiments where we wanted to show that there is not a dependency of number of runs on global protein FDR. Then moving on to the second goal, computational performance. So yeah, there, again, yeah, we significantly improved the speed of Spectronaut 19 with direct DIA. So we already had some improvements that we pushed with version 18.7, but now with Spectronaut 19, you also get a roughly 50% improvement in speed for timsTOF platform. And also, this is really exciting that we really improved the amount of RAM, the scalability of your experiments. So as you can see, you have like 90% less growth in RAM. And we did an experiment where we analyzed 3,000 DA positive runs, and it took like less than 200 gigabytes of RAM. And you could, based on our calculations, you can do a 10,000 direct DIA experiment and be less than like roughly half a terabyte of RAM. There's more, particular aspects to this, but you can reach out to us to find out more about it. And we also improved the amount of space, the scratch space that Spectronaut uses to do your analysis. So yeah, overall, Spectronaut is lighter and faster. There's also more advanced things you can do, so like parallelization to really speed up your performance to get your results faster. So we have a poster, which I forgot to put here, with AWS, where we show using parallelization. You really can get your results 80% faster. Also, like for people who use timsTOF, we have it in Bruker Proteoscape, where you can really seamlessly synchronize your acquisition with your analysis, so you can get your results faster as well. And finally, the third goal, which is candidate discovery. So going back to the 60 data sets, if we look at how Spectronaut 19 performs, on average, we get 10% more candidates. And if you looked at an average result, the candidate curve of this is how it would visually look like. As you can see, yeah. So overall, I think we did a good job with meeting our goals. I hope you guys agree as well. And now I want to very quickly go into some of the nice-to-have things that we worked on with Spectronaut 19. We really improved the label DI workflow in Spectronaut, or Plex DI workflow. You can now get Q values at a transition group level or channel level. You can also do condition setup at the channel level as well, along with your run-based channel specification. We also improved the support for many of these labels in our AI models. And then we also, one of the strengths of direct DI is the PTM analysis, because direct DI is rooted in spectrum-centric analysis, so it doesn't suffer from the same issues that you have with very large search spaces as with peptide-centric searches. We really want to build on this further. So with Spectronaut 19, you can now, for example, normalize, like you can decouple your PTM site quantity from protein abundance with input normalization. And you can also get occupancy rate calculations. So there's a lot more about this, so feel free to reach out to us. And then I hope you all attended the DI seminar yesterday, where Chris had a very great presentation of diagonal positive. It's a really cool new acquisition method. I'll just point you to go, stream it if you didn't attend to understand more about it. But Spectronaut 19 works very well with that. And finally, but not the least, we also have this new deep learning model, which which basically corrects the interference using AI. It's an optional setting, so none of those things I showed right now are based on this. And there's a talk by Anfi where we'll explain on Thursday. So again, can recommend to attend that talk. So we have many, many more features, but not enough time. Come to us at the booth, or say hi and talk to me or anyone else, or just refer to release materials to find out more information. And with that, I would like to thank the entire R&D team at Biognosys, and I would also specifically all the people who were involved more directly with the development of Spectronaut 19. Thanks.
Thank you very much, Tejas. And now I would like to invite Josh to the scene.
Okay, I just needed to hit the switch. All right, good morning, everybody. Thank you for coming. It's really nice to be here with you. And thank you to Monica and the team for giving me this opportunity. Oh no, so there's timing set on this, but I never use them, and since I didn't set this up, we can fight them the whole time, or if there's an expert in here, you can take the timings off. I'll deal with it. Okay, so sorry about that. So thanks to Monica and the team for inviting me to come. And I'm a late arriver.
Important to you. Do you want to?
Well, if you can do it, yeah, I'll keep talking. Yeah. So while he's doing that, I'll just sort of explain. So I'm sort of a latecomer to DIA. We have been practitioners of DDA for a long time. And the title was DIA is the new gold standard, and I don't, I sort of just my perspective as someone who's come around to this concept, and I'm going to try to use this 15, 20 minutes to explain my thinking and how I came to that, and I think many of you are already there. That's why you're in this room. So looks like we're all set. All right, so this is a fun slide that just sort of looks at the prevalence of publications of different data collection methods over the years, and you can see that data dependent has been around for a long time. It's how I learned to do proteomics, and maybe many of you. And you can still see it's the most predominantly used method, but not by much. And as we all know in DDA, we're basically selecting individual peaks and isolating them for MS2. And we can work our way through a chromatogram. And in this case, I'm showing you a variety of different peaks that were selected, but you'll see they don't all get selected, and so you hope that they do. But in about 2014, when we had the first orbit trap tribrid system, when you had the speed of the ion trap systems, you were able to collect, in this case, about 22 tandem mass spectra per second in a data-dependent way, and you can see the majority of these in this example in that 1 second got mapped to sequence. It seemed really good, and we were able to use that technology to map most of the proteins expressed in the yeast proteome in just over an hour, and that was all enabled by a single-shot DDA method and by the orbit trap tribrid platform. And so what I wanted to do, so we've kind of been thinking in that way for quite a while. And more recently, we used basically that same approach on relatively newer generations of the tribrid, but same kind of acquisition strategy to do human cell lysates. And in this case, I'll just walk you through just to give you some comparison to where we're coming from. But in this experiment, we looked at 800 human cell cultures, each using CRISPR to take away a different gene. And we're able to make a lot of measurements. But it took us a little over 3 months to collect just the proteomics data that we're focusing on. And in this case, it took us about a half a year to search that data. So while one can do and could do this scale of project, it's not straightforward, and it's a considerable investment. In that case, we're using 2-hour single-shot DDA methods on that tribrid platform, as I mentioned, and we could get pretty good depth, 8,500 proteins per experiment, and over 5,000 of these proteins detected in all the samples, and 8,400 in 60% of the samples. So the data was good and pretty high quality, and we're able to find new biology, and not just, it's not the point of this talk, but just to say, in this case, we figured out this gene is a part of complex 1 and a CoQ chaperone, and we're able to connect it to human disease, and we made many other insights from that particular data set. So that's kind of where I was at with how to do proteomics. But as you see from the chart, the popularity of DIA is continuing to rise at a very high rate. And in my view, and this is not intended to establish priority, but just sort of in my view of what shaped my thinking in DIA. And there are two sort of innovations. And this paper from the Biognosys team in 2017 was something I took great note of. At the time, I wasn't fully on board with it, but I think it was really, for me, interesting and novel ways to think about this problem and getting really exciting results. And that comes right about here. And so we weren't ready to adopt at that time, but it's something that sort of put me on notice that, hey, there might be something here. And then the second factor that sort of reshaped our thinking was the Orbitrap Astral system that came last year, which has quite fast MS scanning. And so for us, the combination of the tools to be able to look at the data and the instrumentation that has the speed to collect this was pretty important. So you're probably familiar with some of the performance characteristics of that instrument, but my team got invited by Thermo to come check out that prototype Astral in the fall of 2022, and so one of the first things I did was to write Lucas and say, we'd like to get licensed to your software because we're going to be getting some DIA data. So now I'll just share with you some of the results we've gotten with the combination of Spectronaut and the Astral. So we typically run the system like most do for the Astral system for DIA, where we have pretty narrow DIA bins of 2 to 4 Thomson with a 5 millisecond time, which gets us close to 200 hertz, and we're doing MS1s in the Orbitrap. So now if we just might think back to that slide I showed you 10 years ago, we're super excited. In 1 second, we got 20 peptides. Now in 150 milliseconds, we're getting 38 peptides. And you can see some of these scans with Spectronaut are giving us multiple identifications, not surprising given how it's working, but it's just quite a transformation in what we can do. So I thought that, so I have the group now, many of them are doing DIA and they're working with Spectronaut, so I asked them, what are the things you like about this tool? And here are some of the things that they told me. They like this customized report generation, which helps them from making tables manually. And this is a nice feature. They also really enjoy the ability to do in-depth inspection of raw data, and they can look at spectra and look at data quality very easily. And they appreciate this feature where they can do a quick initial evaluation of results. So these are different experiments, and they can look at protein group intensities very easily. So it's been quick adoption and easy adoption because we've only had it for a little over a year. So now I'll show you some of the data. So this work was done by Leah and Trent, shown here. And this is looking at that same cell line that I showed you with the DDA method with the DIA plus on Astral plus the Spectronaut. You can see that getting close to 10,000 or a little bit over proteins with 15, 30, or 60-minute gradients. And the main thing that improves here when you go to longer gradients, so you get a little bit of boost in IDs, but you get better quantitative performance. You can see the CVs are going down, and that's because we're getting more points per peak. So we tend to run 20 to 30-minute gradients this is the area where we do most of our work now. So if I compare to that earlier work from a few years ago where we did DDA, it took us 120 minutes to get 8,600 proteins. Now we can get quite a bit more in a quarter of the time. So it's really remarkable what we can think about. I'm going to skip this one. Now I'll talk to you a little bit about phosphoproteomics, which is something we've had a chance to look at pretty extensively with this combination. So it's a pretty standard workflow with the data collection basically the same as I just showed you, with Spectronaut on the back end. Here's a comparison of tandem mass spectra collected either in the Orbitrap or in the Astral analyzer. You can see they look quite comparable. This phosphorylation work was done by Noah and Pavel, shown here. This is really exciting. So we don't really lose much performance when we go to lower loads, even down to 250 nanograms. And even at sub 100 nanograms, we're getting quite good performance, well over 20,000 phosphorylation sites. This is with a 30-minute gradient. Been very impressed with the overlap and the reproducibility. So you can see that The darker the color here, so the triplicate runs, everything under this bar was seen in all 3 of the runs. This is 2 of the 3, and this is 1 of the 3. So quite good data completeness. And here's another example with phosphopeptides of the CV values for technical replicates coming down as you have a longer gradient, right? So that's the trade-off. So we needed to figure out how good the instrument and the software could localize sites. And this is an experiment was inspired by Jesper Olsen's publication a few years ago, but basically what they did was they took yeast phosphopeptide background and synthetic phosphopeptides. In this case, we spike in, and then we run the system and we try to localize, we figure out the FDR, and we figure out quantitative performance. So here's some of the results, and these data that I'm showing you in the blue bars are with Spectronaut 19. And so what we're seeing here is the probability cutoff, and the dark blue is the Orbitrap Astral data, and then this is Jesper's data that was originally reported, and then we researched it with Spectronaut 19 and see very good agreement with the prior results. And then here you can see the average number of correct precursors that we get as a function of the localization probability cutoff. Now, I said there was 225. We don't get them all because some overlap with yeast protein peptides, and then a few we don't get. But you can see we're doing pretty good. Even at the very stringent cutoff, we're getting the vast majority of these included. So we wanted to look at quantitative performance, and so we can spike these standards into the yeast matrix background at different amounts, and we can see how linear the values come out. And you can, if I could just look at this one, you can see the 5 different spike in values. We get very good linear correlation with the amount loaded, and quite good R-square value. So these are 3 randomly drawn peptides of the 225. This chart shows you the aggregate performance. So R-square value here, you can see most of them are well above 0.95 and close to 1. And we had a few low performers, but most of them up here. And then again, we looked at data completeness and triplicates, and you can see for even very low loads of the 39 attomole, we're getting about half of them detected all the time. So I was really impressed with the ability of the Astral and the Spectronaut tool to pull these out consistently and correctly. And then these plots are interesting because they show us how well we modeled with this range of concentration the actual sample. So the yeast background is in black, and you can see for the high load, we're modeling the high abundant stuff, and at the lowest load, we're doing a good job modeling the low end of that distribution. So we felt like those numbers are a good range of what we would actually see in a real sample. And then this data shows us that we wanted to do a real experiment and to compare how well this worked across many platforms and with Spectronaut. So Matthias Mann's group published on timsTOF with the HeLa cells stimulated with EGF or not. And so since we didn't have one of those systems, we replicated this as best we could. And then we replicated those conditions. And we ran that on the Astral with DIA and the Ascend with DIA. And then we researched all of the data using the library-free method of Spectronaut 19. And the point here is that the blue things are things that should be going up in stimulation. And I think what's really exciting is no matter how you collected this data, which platform you use, you're going to find good biology. And so I think that's really a testament to the software and how well it's working and to the method. So finally, and we had done some work with mouse and wanted to compare how much have things advanced since this really fun paper from 2010 from Steve Gigi's group, where they took many tissues of mouse, did deep fractionation, data-dependent phosphorylation analysis. And in our case, we used the combination I just mentioned, but we only spent a half a day, about an hour per tissue. So we had 12 tissues. And it's really remarkable whether you spend about a month or a half a day, the difference that 14 years can make in the technology and the number and the depth and the speed of sites that we can get at. The final project that I want to talk about is led by Meg Robinson in my lab in a collaboration with Johan Owerks of EPFL. And Johan has created a mouse model of NASH or MASH, this liver disease. And he has taken 4 strains of mouse, and he's in and outbred them to make 500 F2 animals that have essentially shuffled genomes, you could think of. And then they genome sequence all of these animals. And what we want to do is take the liver, and we're going to take from the 500 animals, and we're going to do liver proteomics. And the idea is that we will ultimately do QTL mapping. So I'm showing you any quantitative trait, and in this case, just for simplicity, we're going to think of body weight. So all these 500 mice have a mass, and we can just weigh them, and you can see a distribution in body weight. And then you can take that quantitative value, and since you know the genome sequence of all the animals, you can try to correlate that to specific gene loci. It's called quantitative trait loci mapping. And we're going to do the same thing with every protein value, except we'll get 10,000 protein quantitative values, and we'll be able to map the expression of every protein to a genomic loci. That's the idea. But first, we have to get that result. So Meg took the 500 plus a bunch of control livers and did a 30-minute method, basically what I just showed you. And she was able to get nearly 10,000 proteins per sample on average with that liver tissue, about 6,000 proteins in all the samples and 9,260%. So performance very similar to what I showed you earlier from cell lines, but what I would argue is a tougher matrix. And she did it in a quarter of the analysis time. So it took us a month to collect that data. But I think for the purposes of the data search, it's truly remarkable. It took 3 days with Spectronaut 19 to search this data, nearly 600 files that were very large, compared to what we had experienced before with the DDA data. And in fact, she told me yesterday, or just before we left, she said, well, actually, I started the search on Friday morning, and I came in on Monday, and it was done. So it might have been less than 3 days, but very fast. Super incredible. All right, so I'm going to wrap up now. I started with this slide. These are my thoughts about how things are going to unfold, but I think that MS scan rates are doubtless going to continue to rise. They're going to get faster. That is going to mean that we will be using shorter gradient lengths. So gradient lengths will fall to less time. So we might have very deep analysis that we're getting now, but we'll get it in even less time. I note that this is what I think will happen. And the software innovation that we've seen this morning will continue to improve our ability to quantify and increase our coverage with that data. And so I do fully expect that the DIA method will become the most prevalently used because of the nexus of all these things that are happening right now. Well, here's the team. I highlighted most of the folks as we went along, so I won't do it again, but I really want to mention I'm grateful for support from the National Institutes of Health, both of these grants here, also from Thermo Fisher Scientific, the Morgridge Institute for Research, and the University of Wisconsin. Thank you for being here this morning.
Thank you very much, Josh. Great talk. Please, if you have any questions, hold them till the last talk, and we will have this common Q&A. And now I would like to invite our last speaker of today, Denys. Please welcome.
Yeah, thank you guys for inviting me over. It's really great to speak to you today. Yeah, so I wanted to talk to you today about how we think the high-dimensional phosphoproteomics can work. Yeah, I would like to switch gear in comparison with the previous presentation and talk about like a little bit different family of mass spec instrumentation. So timsTOF is the pretty fast mass spectrometer with an added advantage of the ion mobility in this very small but very powerful TIMS strapped ion mobility spectrometry device that allows us to separate peptides and phosphopeptides additionally to the liquid chromatography dimension in the gas phase, which allows us to record an additional dimension of the data, the ion mobility, and in the end, the output data looks like this in the 2D coordinates, where we can separate our peptides and phosphopeptides both by mass and ion mobility. And if you can see the separation of peptides and phosphopeptides looks a little bit different. So prior to setting up the workflow, it is very important to first, prior to do the DIA, it's very important to do the DDA to actually find out how these coordinates are looking, and then to optimize your DIA windows accordingly to maximize the output. But the timsTOF instruments are very fast, so what we can do with it in the context of the phosphoproteomics, so the first things first, we can increase the throughput, and this graph is something that you may have already seen in the previous presentation, but in slightly different context. So here we didn't go with the lower amount of injection material, but with the shorter gradients. And as before, we also saw that the amount of the number of identified phosphopeptides doesn't really decrease with fourfold decrease of the gradient time. This is partially thanks to the ion mobility, because when you go shorter in gradient time from 60, let's say, to 7 minutes, you obviously compress the peak shape quite significantly, but the resolution in TIMS is not dependent on the gradient time and is constant, which adds a constant number of the constant degree of resolution regardless of the gradient, which also helps us to decrease the complexity of spectra that we acquire as we go through shorter gradients. And with ion mobility, we are able to quite substantially decrease the number of co-isolated and co-eluted features that allows us to identify more phosphosites with better quality. But this is all good. The instruments are pretty fast. The software is working nicely, which means that the next bottleneck for the high throughput phospho research is actually how we deliver our phosphopeptides to the mass spec, meaning that the sample preparation workflows should be also optimized up to date in both in regards of high throughput and high sensitivity to actually match the capabilities of the instrument. So with this in mind, and with help and advice of Sean Humphrey, we created, on the base of the EasyPhos phospho enrichment platform, we created something that we now call the MicroPhos, where we specifically designed it and optimized it to support the high throughput 96-well plate format from the cell culture, so from cells to phosphopeptides, how we like to say, and as an added bonus, by decreasing all operational volumes to actually support the high throughput 96-well plate format processing. By decreasing the operational volumes, we are increasing the sensitivity of the enrichment quite significantly, as I will show a bit later. But decreasing the operational volumes comes at cost. In this case, the cost is the quite significant increase of unspecific binding of the peptides, because as you can imagine, the more you increase the concentration of the peptides in the limited volumes, the more unphosphorylated peptides will bind. And to overcome this issue, we performed a quite thorough screening of potential selectivity agents. And at first, we didn't go anywhere because neither of the selectivity agents couldn't perform very good or optimal in our reduced volumes alone. But quite fortunately, we stumbled across the combination of several selectivity agents. In this case, it's the combination of the glycolic acid and potassium monophosphate, because, for example, here in the selectivity, if you just increase the concentration of the glycolic acid, you do not really get to the highest percentages of selectivity, meaning there is still quite some portion of the unphosphorylated peptides in your solution after enrichment. And if you, for example, just increase the concentration of the monopotassium phosphate without adding the glycolic acid, you achieve pretty good quality of enrichment, but with the decrease in the relative fraction of the phosphopeptides, meaning that too high concentration of monopotassium phosphate actually is bad for the number of identifications. But if you combine them, you are reaching this very nice and very wide plateau of optimal performance, both in terms of selectivity and the number of identifications. And the added bonus here is that even if you mess up a bit your pipetting, so plus minus 0.5 molar glycolic acid or a couple of millimolar error of the monopotassium phosphate will not drastically change the quality of your analysis. And then this is not the only thing that we optimized. We also optimized a lot the protein digestion by significantly reducing the digestion from overnight to just several hours. And in comparison with the previous generation EasyPhos, we are actually now able to perform all steps of the experiment, so from cell culture to the clean phosphopeptides in just around 8 hours of the working time, plus of course extra mass spectrometry time. Then the better, faster, more sensitive instruments really help to increase the number of identifications. For example, the timsTOF HT, with which we performed the majority of our optimization experiments, when we compared it to timsTOF Ultra, the raw intensity of identified class 1 phosphopeptides increased quite significantly, especially for the quite low amount of starting protein material, so for 1 microgram. And with timsTOF Ultra, we were then able to identify reproducibly more than 30,000 unique phosphosides and around 16,000 of class 1, so significantly quite with the localization probability of more than 75%. And if you compare the upper part of the graph and the lower part of the graph, so the lower part of the graph is the dilution experiment that we made. So we enriched first enriched the phosphopeptides from 20 microgram of starting protein material and diluted it to 10, 5, and 1, and then did the same but took 10, 5, and 1 microgram of starting protein material and enriched from that. You do not really see the difference, meaning that the quality of the enrichment doesn't really drop on this quite wide range of the starting protein materials, meaning that we can actually deliver the vast majority of all phosphopeptides from the cells, even from just a couple of thousands of cells to the mass spectrometer for the analysis. And well, this analysis was done on the Spectronaut 18. The Spectronaut 19 actually increases the number of significantly localized phosphopeptides over the very wide area of the enrichment. So, for example, we here can, from just 62.5 nanogram of starting protein material, we can identify a little bit less than 1,000 of class 1 phosphopeptides. And also here I have to note that with the when we go in lower with the starting protein material, we actually advocate on using a little bit higher localizational probability thresholds for at least 0.9, so at least 90% to actually be very sure that these are the phosphopeptides that you are identifying, that they are on this exact spot. And we are identifying not only a lot of phosphopeptides, we are also identifying a lot of functional phosphopeptides, so those that really play a significant role in the life of the cell or tissue. So, for example, in this experiment with EGF, with the EGF treatment of HeLa cells, we concentrated our attention on so-called blottable sites, so those sites that are frequently and currently used in the Western blotting analysis. And we were able to identify the vast majority of those from 1 microgram to 20 microgram of starting protein material with quite equal localization probability of more than 95%, meaning that now even starting from much less material than from Western blots, we can identify tens of thousands of phosphosites with very high localizational probability. And if you guys don't like the manual sample processing and like to optimize your automation, this is also very easily automated on the automation platform of your choice. So here we show it for the SAMAP Bravo with the titanium O2 cartridges, which further decreases the time of preparation to roughly twofold. And with this, we are able to identify very equal amounts between the. So if we compare between the automated and the manual approach, we are identifying roughly similar amount with numbers of phosphosites with the very similar quality. So this platform is you can also automate it without any or major problems. But this is also all good. We can now identify a lot. We can identify functional, but how to apply it? How do we apply? So the first area that we thought of is actually the high sensitivity tissue phosphoproteomics. So for this specific case, we are excising the very minimal, very minuscule amount of tissue. So in this regard is the subregions of the mouse hippocampus. And by analyzing this data with Spectronaut, the latest Spectronaut with Spectronaut 19, we are able to identify separately for each region. So if you analyze each of the regions separately and not together, you can, for almost all of them, identify more than 20,000 class 1 phosphosites and achieving the vast coverage of the mouse brain phosphoproteome with the very nice reproducibility and quantification precision. And already here, from just very minimal amounts, we are able to cover the vast majority of the GPCR receptors C terminuses, and with high degree of reproducibility to identify and quantify a lot of serines, threonines, and serines and threonines on the C-terminuses of GPCRs that play a major role in determining the cell fates, the cellular fates upon the GPCR signaling. Another area of implementation, and actually the high-dimensional phosphoproteomics, is to take the full advantage of the throughput and sensitivity, because usually the phosphoproteomic approaches take one perturbation at a time, single dose, single time point, and compare them, a control versus treatment, let's say. What we do here with microphos is the highly parallel cell culture in the 96 well-plate format with different time response, with different time responses and different doses of drugs, and all which all is done in parallel in the area of one or several 96 well plates, which actually helps to save a lot of costs for the very high throughput ideas that you may have in mind. So to show how this works, we did the time-dependent treatment for the four most prominent and most highly used tyrosine kinase inhibitors that are used in clinics for treatment of the chronic myeloid leukemia, so against the BCR-ABL fusion protein. We incubated the BCR-ABL mutated cell line in the 96 well-plate format for 4 time points, and then analyzed the 192 samples of both proteome and phosphoproteome with the microphos platform, and subjected them to our optimized phospho-DIA analysis with the timsTOF, and we were able to reproducibly across all conditions to identify more than 20,000 class 1 phosphocytes just from a couple of millions of cells to start with, but with 16 different conditions. And by performing the area under the curve-like analysis for the time courses for each drug, we were able to identify a very intricate, very small differences between the drugs with generally very similar mechanism of action, but we were able to identify some differences in the phosphorylation levels of different histone deacetylases, and now we're currently continuing working on this experiments further. Yeah. So to sum up, I would say that fast instrumentation and very quick, very quick and very reliable analysis with the Spectronaut, and high sensitivity and high throughput sample preparation, automated or manual, we are now able to achieve a very high throughput phosphoproteomics that opens a lot of very exciting, very cool applications that we are envisioning in the next couple of years. And of course, all of this wouldn't happen without all these very cool people, and especially my boss, Florian Meyer, who is a very nice. Unfortunately, he couldn't be here this year, but yes, thanks to all of them, I'm able to present all of this data to you. And of course, yeah, thank you for your attention.